everyone, thank you for having me. I hope you can all hear me and see my slides. So my talk will be about the cardiovascular system. I am currently a third-year PhD student at the University of Oxford, and my PhD is in the cardiovascular sciences. So just a brief outline of what I'll be talking to you about today. First, I thought we'll do a rough introduction to the cardiovascular system, some anatomical structures, and mostly focusing on the circulatory systems. Then focusing a bit on the cardiovascular diseases, and there I'll talk a bit about my research as well. Then I tried to come up with an interactive study to explore the coronary vascular disease risks. And finally, to make it Oxford specific, I will be highlighting a lot of research excellence that is going on at the university. And afterwards, you'll have time to ask any questions that you might have, but also feel free to put them in the chat if you've got anything in the meantime. So I have put a few activities in there for you to engage with. So I thought I would start with asking this. If you could fill in the gaps of these sentences with what are the missing structures of the cardiovascular system? I don't know if anyone's put anything in the chat, but so far I can't see anything. Is that right? Oh, yes, someone has. Yes, it's heart, the first one. The cardiovascular system consists of the heart, that is the muscular pumping device. Then we've got arteries, veins, and capillaries. That is correct. So the cardiovascular system is a full organ system that transports blood throughout the body to all tissues, delivering oxygen and nutrients. And then it also collects carbon dioxide and other waste products and delivers it back to the heart. It is a closed system, starting with the heart and then also ending with the heart. The two circuits that the heart pumps the blood into are called the pulmonary circuit and the systemic. So a huge question is, why do we still need to study the cardiovascular system? It feels like we know everything that is the heart and the vessels. Why is there still a huge need to study it? So I wanted to ask if you happen to know what is the leading cause of death in the world currently. Yes, it's not a big surprise considering my talk that it is actually coronary heart disease. Coronary heart disease is still the leading cause of death for both men and women globally. If we look at UK specific statistics, coronary heart disease is actually the second leading cause of death after Alzheimer's and dementia, but still a massive burden on our healthcare system. Every 8 minutes, someone in the UK dies from coronary heart disease, and there are over 2.3 million people living with the disease currently in the UK. So the heart is a muscular pump that provides the force to be able to transport the blood throughout the body. It has four different chambers that, before looking at the graph and peeking at it, I was wondering if you guys already know what are those four chambers and what type of valves are present within the heart. We have, yes, we have atrium and ventricle. We do have the right and left ventricles, and we have the right and left atria. The valves Oh yes, someone already has put them in. Atrioventricular and semilunar valves. So looking at these chambers, if I change to a laser pointer, we do have the right atrium and the right ventricle, the left atrium and the left ventricle. Considering the valves, the atrioventricular valves are the ones present between the atria and the ventricles, and the semilunar valves are at the base of the large vessels. In the blue, that is the pulmonary artery. You can see the valves there, and at the base of the aorta, you can also see them. So the heart, to have this massive force to be able to pump the blood, needs to have a very thick wall that is composed of 3 distinct different layers. Looking from the outside of the heart in, the outer layer is called the epicardium. Then the middle layer is called the myocardium. That is where you've got all the muscle present in the heart tissue. And then the inner layer of the heart is called the endocardium. Now the heart is surrounded by another layer, which we call the pericardium, which is a sac that covers the whole heart. I've put some fun questions in there, which I just wanted you to guess if you know how much blood does a healthy adult heart pump in 1 minute. Yeah, we have one correct answer, and that is 5 liters. And if you were to take a guess at how many times does a heart beat in the average person's life? Oh, wow, we've got brilliant students here. Yes, it is indeed 3 billion. And how long would it take for a red blood cell to be circulated through the whole body? 2 minutes is a bit too long. Yes, it is indeed 20 seconds, very quick. So I just wanted to talk a bit about how the blood actually flows throughout the heart. What is important to know that is you have to think of the heart as the upper part and the lower part. The upper part, both atria contract at the same time, and the bottom part, the ventricles also contract at the same time. First the atria, then the ventricles. So if we start with the right atrium, then the blood flows into the right ventricle, here in blue, and then once the ventricle contracts, the blood goes into the pulmonary circulation through the pulmonary artery. This is deoxygenated blood that is transported to the lungs to be oxygenated. Once that blood is oxygenated, it flows back into the left atrium. From there, once that contracts, it goes into the left ventricle, and once the ventricle contracts, the blood goes into the aorta and the systemic circulation. So the two circulatory pathways are the pulmonary and the systemic. The pulmonary circuit is where the blood gets oxygenated. It starts at the heart, goes to the lung, and back to the heart. The systemic circuit provides blood to every other tissue in the blood. Actually, it provides blood to the lungs as well, as in supporting the lungs tissues with oxygenated blood. So they both carry oxygen and waste products, but in the opposite direction from the blood, from the heart. Yes. So as a vascular biologist, my main focus is the vessels, and my main interests are the vessels of the cardiovascular system. So vessels are tube-like structures that carry the blood. They deliver the oxygen and nutrients and also remove the waste products. So arteries are often defined as the vessels that carry the oxygen-rich blood away from the heart. But if you think about it in case of the pulmonary circulation, that does not stand because the pulmonary artery actually transports oxygen, low oxygen per blood to the lungs. And the veins are the ones that would return the oxygen poor blood, except for, again, the lung. The most important, bit biased, but the most important vessels in the vascular system are the capillaries. They are the surface for the exchange of oxygen and the nutrients. And as a cell biologist, I think this is a very important part of the vessels, and it's knowing the structure of how the vessel walls are made up and how different they are if you talk about different vessels. Focusing mostly on the larger vessel types, they often have the three layers called the intima, which is the innermost layer. It's usually a layer of endothelial cells. Then you've got a basement membrane that separates them from the media, which in arteries is mostly a very muscular, full of smooth muscle cells and some connective tissue. But in a vein, that layer is, as you can see, a bit thinner and less muscular. And the outer layer is called the adventitia. So if you think about it, this difference between arteries and veins are very important depending on where they transport blood from and to. And that's why arteries need to be a lot more muscular to be able to regulate that vascular tone and keep the blood flowing. And if you look at the smallest vessels, the capillaries, they are usually just one layer of endothelial cells, and they are often supported by different cell types like pericytes. So this was just an overview of how things are structured and how things are working when they are working, but unfortunately, they don't always work the way they should and the way we wish they did. So I wanted to talk a bit about cardiovascular diseases. And it's important to note that majority of cardiovascular diseases originate from endothelial injury. So that is the injury to the innermost layer of the vessel walls. So this injury can be a huge variety of things, such as inflammation, different types of bacteria and viruses causing inflammation, or it can be damage to the vessel wall and the consequent inflammation. That can also lead to apoptosis and necrosis, so death of cells within the arteries causing this. Oxidative stress and reactive oxygen species often cause endothelial injury as well. And mechanical injury, such as the shear stress, so that often happens at the bifurcation of arteries. So once an artery branches into two, that's usually an area for high shear stress, and that is a very atherosclerotic prone area where we often see plaque development. So the underlying cause of a lot of heart diseases is atherosclerosis, and not just heart disease, because this can happen in any vessel really in the body. And it is a process where there is a plaque buildup in the arterial wall. And the most serious cases is when there is a blockage or when there's a rupture, and that plaque that was built up in an artery travels through the circulation, and when it reaches a point where it can block an artery, that can cause strokes in the brain or myocardial infarction or heart attack in the heart. So I wanted to show you a real world example of how we study this, how we study cardiovascular diseases and how we study atherosclerosis, both in the sense of basic science and then applied science translation to real world application. So when you think about research and studying the cardiovascular system, what tools do you think we have of this list that we can utilize in a research setting? We've got one answer. Animal models. Yes, we do have various animal models, and they are very commonly used in cardiovascular research. Yes, we have all of them, which is correct. We do utilize most of these. Animal models, cells are very commonly used for cell biology. Postmortem tissues are often the most accessible human tissues we can get, donor tissues as well. And clinical observations, imaging, that is very useful and a functional readout. And that is one setting where we can access healthy individuals to study to have a comparison between healthy and disease. And it becomes a bit tricky when it comes to healthy heart samples, because if you think about it, healthy hearts are not really accessible in the sense you don't send a healthy patient surgery to get some samples from their heart. It just doesn't happen. And if we have a heart, a donor heart, that is from a healthy individual, that would usually get transplanted, rightfully so, because that can save someone's life. So it's really uncommon to have healthy heart samples and healthy heart tissue accessible to research. It's very rare, and once you have some, that can get you a major publication just on its own. And coming a bit to my research focus, I wanted to ask if you guys know, based on all I've talked about as well, what is the most abundant cell type in a vessel wall? And mostly thinking of not capillaries, but a bit larger vessels. Yes, it is smooth muscle cells indeed. Smooth muscle cells are the most abundant cell type in the vessel wall, but because I mentioned that a lot of the cardiovascular diseases originate from the endothelial cell layer, most of the research is focused on endothelial cells rather than smooth muscle cells, which are the most abundant. So I wanted to introduce a bit of how we can model cardiovascular diseases in a dish in vitro. So the way my work works is I access human primary tissues. So that means it's tissues that come straight from a donor. It's usually, as with heart samples, a donor is someone who passed away and they or their family donated their tissues. And from those tissues, companies isolate cells, and that is accessible for researchers. You can actually purchase vials of human cells for research purposes. So that is what I do. And because I'm trying to model a disease where I induce the disease, I'm aiming to start with a kind of healthy phenotype in the model. So that means when you're purchasing cells, you have to factor in information about the donor, such as age, ethnicity, obesity, and habits such as alcohol consumption, smoking. So once you've got these cells, I wanted to show you a phenomenon that happens in disease, specifically in atherosclerosis, where we start with spindle-shaped smooth muscle cells that are present in the coronary arterial wall in healthy individuals. This is how they look like. They're very organized. They form a monolayer. They're quite close together, and this is their usual shape. But in disease, sometimes they undergo a shift in their phenotype and they become this rhomboid shape, so-called synthetic phenotype. So I hope you can see that is a very striking difference of how smooth muscle cells can look in disease. So this phenomenon is called the smooth muscle cell phenotypic transition, where we start from a contractile, healthy smooth muscle cell that can de-differentiate to a synthetic phenotype. So it's a de-differentiation because in the developmental process, smooth muscle cells do undergo this intermediary stage through their differentiation. They become synthetic and then contractile, showing in the green arrow. That is a developmental process. But in disease, this de-differentiation can happen, which we wouldn't really want, because once their contractile smooth muscle cell phenotype, they're where they are. They're there because they have to be that. You don't really want them to lose their phenotype and their function in that setting. So I wanted to ask if you could think of consequences this transition could have. What would happen to smooth muscle cells that change from this contractile phenotype to a synthetic phenotype? I don't know if the questions were too easy or you just all are brilliant, because yes, it is all of the above. The cells do lose their contractile power, and therefore they don't have the ability to regulate the vascular tone to the same effect they did before. And they also contribute to the formation of atherosclerotic plaque in the arteries. So why this happens? It also happens in response to endothelial cell injury. So that is your starting point. And due to that injury, so these cells are immune cells. They are macrophages, platelets, and they secrete growth factor called platelet-derived growth factor B. So those yellow dots are those secreted PDGFB molecules that are secreted by all of these immune cells and the damaged endothelial cells. So that reaches the level of the smooth muscle cells in the vessel wall and causes them to enhance their proliferation and migration. And they migrate from the media where they normally are into the into the intima, to the inner layer of the vessel, and that causes a whole lot of issues. It destabilizes the vessel wall, it disrupts the vascular tone, and also constricts the vascular wall, both because there's more of them, but also just being there. So it's really not something that you would want to happen. And in the context of an atherosclerotic plaque formation, the smooth muscle cells are the ones highlighted here. It's not a black and white kind of they're all bad guys situation, because in a plaque, it doesn't just matter that whether you have a plaque, but how stable that plaque is. Is it prone to rupture? Is it gonna rupture and then cause ischemia elsewhere? Or is it a stable one that is just there, still not good that it's there, but it's stable, it's not gonna cause huge issues. So one factor that can stabilize the plaque is a smooth muscle cell layer on top of the plaque, which we call the fibrous cap. And the orange part would be the necrotic core. That's a lot of inflammation there, a lot of dead cells. But the role of the smooth muscle cells in this formation can be good and bad. As I mentioned, it's good that it stabilizes the plaque, but also that is one of the initiating factors of plaque formation. So why is it good they stabilize it? But had they not migrated at all, maybe you wouldn't even have a plaque. So that can be a good and bad thing as well. Also, as they become synthetic, they can acquire different phenotypes with different functionalities they would not have as a contractile smooth muscle cell. For instance, they can become fibroblast-like, and they can secrete an extreme amount of extracellular matrix, contributing to the increased size of the necrotic core, and they can also become macrophage-like, inflammatory, contributing to the inflammation in the necrotic core. So I would say it's mostly a bad thing that they do lose their phenotype, even though there are some advantages to the stabilization. So, how can we study this in a research setting to identify targets, therapeutic targets, that could inhibit this transition and inhibit plaque formation? So I start with the cells I purchased. I grow them in plastic dishes, really. And then you can use the growth factor that we know in vivo in the humans is a causal causer of this phenotypic transition, PDGFB, platelet-derived growth factor B. And that's how I induce this transition we see in human samples, in tissues, in cells as well. And then I have a huge array of tools I can use to investigate what happens to the cells. So one of them is imaging. We've got a lot of microscopes. These images are bright field images. So you can use this to look at the morphology, the cell shape changes, which you know is associated with the disease. So you can see the clear spindle shape, healthy control cells, and once they get induced, you can see how they transition into this rhomboid synthetic state. Then, not going into huge depths, you would want to look usually in cell biology on the gene level, gene changes. And for this, we use a technique called qPCR, quantitative polymerase chain reactions. It's just an assay to look at the level of expression of your genes in response to treatment. So looking at these plots, on the top, you can see the name of the genes, ACTA2, that's alpha smooth muscle actin, and you can see that its level high up in the control, and once you give it treatment, it drops very significantly. Same with this other gene. So these are genes that form part of the contractile apparatus. They form actin filaments on the protein level that are very important for the contractile phenotype. And then we've got these other genes in pink, and they are associated with the synthetic phenotype. And you can see their expression is higher in response to treatment. So that's the kind of changes you would look at on the gene level. Then usually you look at the protein level as well, but you also want to look at the functionality of the cells. So as I mentioned before, we know that with this synthetic transition, they acquire a high proliferative rate and migration. So there are different cell-based assays you can use to look at these functions. For instance, this assay I'm showing here with these fluorescent images are looking at the proliferation of the smooth muscle cells. The top three images are the control, and the bottom three are the ones that had the treatment, the induction of the synthetic phenotype. And basically, this assay measures or looks at the incorporation of EDU. It's a labeled uracil base that emits a fluorescent light. in this case, this red. So that means when the cells are replicating the DNA, this is incorporated in there. So you can tell that the nuclei that formed, because, sorry, I should have mentioned, these little dots are the nuclei of the cells. So we can tell from this, there's been a lot of replication in response to the treatment. In the same time frame, where the control setting, we can see just one replication. And this is what's shown on this plot. Basically, it's just quantifying these images, because usually it's not enough to show the images, you have to quantify it. And it's very easy here because you just count those little dots, as in not me, but like a software. So you can see it's a very significant enhanced proliferative rate of the cells. So this is just to show you how a basic science research kind of would look at coronary artery disease in a dish. Hopefully that gives you an idea of how you can model a disease in a dish, trying to look at a phenomenon we know is detrimental, and we would want to develop targets to inhibit this and hopefully decrease coronary artery disease. So now I wanted to move on to the coronary heart disease associated risks, but you've all been really brilliant, so I'm fairly sure this will be a very easy task for you. But I wanted to just talk about it with you as well. So there was a patient who presents at the clinic, a 50-year-old male, reported smoking, high blood pressure, a family history of heart disease, occasional chest pain, and a shortness of breath during exercise. Can you identify potential risk factors from this medical history the patient has reported? Yes, smoking absolutely is a huge risk factor for cardiovascular disease. High blood pressure, absolutely. Family history of heart disease, predisposition to coronary artery disease is one of the most significant ones. Older age, shortness of breath, you've not mentioned, but that is also indicative of this person living a sedentary lifestyle, not exercising enough, and not exercising is a massive risk factor. Age, a couple of you have highlighted. Yes, age is also a risk factor for coronary cardiovascular disease. And one I have actually not seen any of you mention is actually gender. Gender is also a risk factor. So there were a variety of risk factors in this medical history, and I'm sure you can tell that some of them are out of your control. Those we would call the non-modifiable risk factors. Here we would talk about the genetic predisposition, the family history. Race and ethnicity are also a risk factor for cardiovascular disease, so is gender and aging. So yes, these are things you can't really do anything about, but there are a lot you can do something about. Obesity, diabetes, smoking, alcohol use, diet, exercise. These are all things that contribute massively to coronary and cardiovascular disease development. And this is the first step for prevention. Unfortunately, as you could see from the statistics, we cannot cure coronary heart disease and heart disease. So highlighting modifiable risk factors and prevention is really our current best approach. So now thinking about how you could reduce these risks, what would you recommend to this person who presents at the clinic? We have a question: which ethnicities and gender are more likely to develop cardiovascular disease? Ethnicities, it's actually African Americans are at a higher risk for developing cardiovascular diseases, and gender is not as clear cut. Men are likely to develop coronary disease earlier, but severity is often worse in women. So for the recommendation, we've got exercise, absolutely. Quitting or reducing smoking, yes, probably quitting would be the most ideal. Diet, absolutely. Yes. Yes, all of those are very important. And what someone presenting to a GP with these symptoms would probably be recommended immediately. And lifestyle habits, yes, stress management in there is something. Yes, oh, amazing answers. Stress management, stress is also a huge risk factor, not just for cardiovascular disease, for a lot of other diseases. So practicing mindfulness is quite important. So I wanted to focus the last part of my talk on what is going on at the University of Oxford in the sense of cardiovascular research. So some of you might have heard this in the news that quite a few research centres, but Oxford specifically, was renewed as a centre of research excellence funded by the British Heart Foundation. And this means a cohort of scientists working on trying to research and cure different types of cardiovascular diseases. There's a lot of different types of research surrounding this area, a lot in atherosclerosis, heart failure, regeneration, of course, vascular biology as well. And a lot of our techniques is focused on genetic research using bioinformatics. We do a lot of sequencing. We sequence RNA, single cells, anything you can sequence, we do it. So a lot of multi-omics approaches. And the aim of the center is to better understand how heart diseases develop, how we can intervene, how we can develop treatments. And also a huge focus of the center is on training scientists. So within this center, there are a lot of PhD opportunities and fellowship opportunities. So that would be your next step after your PhD, to develop scientists. And with the center, the approach is to work together as a huge group of scientists. There are, in this graph, you can see the different themes of the center. One focus on drug discovery and delivery. We've got repair and regeneration, and big data and computational science. So bringing together expertise from very different areas and different techniques together, we've got a better chance of achieving the aims of the center. So one cardiovascular research you might have heard of in the news a few years ago now is the Cure Heart project. So to put this into context, this center I just mentioned, the award of funding for research by BHF for this center for the whole of Oxford University is £6 million. A few years ago, Professor Hugh Watkins was awarded £30 million for the Cure Heart project. So that is the biggest grant BHF, the British Heart Foundation, has ever given to one project. And their aim, the aim of the Cure Heart project, is to focus on inherited heart conditions that affect 1 in 250 people globally. So it's a very common genetic disorder. And what they are trying to do in this project is to identify genetic mutations that cause the disease to develop. And if they manage to do that, they could diagnose patients with this inherited heart disease. And I think the idea is genetic editing to prevent development of heart muscle diseases. So I've included in the last slide some links to listen to Hugh Watkins talk about this project and the whole aspiration of it, which is very inspiring, very interesting. Highly recommend you guys listen to it. And another highlight, I would say, of the research that is happening in Oxford is called the Oxford Heart, Vessel and Fat Cohort. So this initiative is led by Professor Harris Antoniades at the John Radcliffe Hospital in Oxford. And they've recruited over, I believe, 1,500 people to participate in this study. And that means they take different samples from the patients and they do different imaging techniques. So they have compiled a huge database of a large number of patients that can be used to study cardiovascular diseases. The focus of the Harris group is on fat in the arteries. So they use this fat attenuation index. And basically, I think the main approach here was imaging and using this as a measurement to predisposition for coronary heart disease. So they can spot this disease early and trying to intervene at a very early stage. Because as I said before, it's mostly prevention we can do at this stage, and it's probably our best shot than trying to reverse the effects of a myocardial infarction. Although that research is also happening at Oxford, specifically Professor Riley's group. Paul Riley is looking at the regeneration of heart tissue after myocardial ischemia, if any of you are interested in that. He is on the slide here, I believe. Yes, Paul Riley. So you can also look that research up. That's also very interesting. So I've come to the end of my slides. If you've got any questions, please feel free to put them in the chat. And just to mention again, I did put some resources in here for you to look at, specifically about the cardiovascular research that goes on in Oxford. There's over 40 PIs, personal investigators. Is that PI? So you can read about everyone's research that is happening at Oxford about cardiovascular disease, some statistics from BHF, and some interactive tools if you want to explore the cardiovascular system a bit more in depth. The Hidden Heart program from the Antoniades group about the fat and using that as a readout. And for the Cure Heart, there are quite a few interviews with Hugh Watkins that I highly recommend.